How do bees become queen bees? As most people know, this happens when royal jelly is fed to the larvae of the bees, and that transforms them into queen bees, which are able to produce eggs and produce new larvae. Without that royal jelly, those larvae just develop into worker bees. The mechanisms of which that happens are DNA methylation and siRNA. To put it in simpler terms, these processes restrict genetic information from being expressed. So, naturally, without any methylation or siRNA or any royal jelly, which induces that, bees develop a gene called DNMT3, which turn off the production of ovaries, which restricts them from becoming queen bees. So, scientists in this paper tested this to confirm if it is true or not, and to maybe develop new ways which we can increase the bee population. Bees play a crucial role in maintaining both human and environmental health. Their honey and other byproducts possess medicinal properties, and their function as pollinators is essential for sustaining food production. Unfortunately, though, the bee population is declining. Using this research, not only can we answer in what ways can queen bees develop, but also open new doors to new research for m- maybe a medicine or a spray that we can provide to the bees that creates new queen bees and raise the bee population.
So I'm here with the methods section, and here um, I wanted to just sort of take the opportunity to explain the control and experimental groups, which will sort of help inform what we need to know for the methods section. So we have the wild type as a control, which is just sort of honeybees you can find out in nature. And we also have honeybees without a methylation protein. So methylation is just a way for the body to control what genes from your DNA are expressed and what aren't. And what these researchers more specifically wanted to find out was how does DNA methylation affect role expression? So basically, what they did was they removed a methylation protein called DNMT3 with small inhibiting RNA. Um, Basically, what that means, there's this protein that sort of overlooks the process of methylation, and the researchers added something to it to make it so, and they made a mutant that didn't produce this protein that regulated methylation. And that's how the researchers studied the effects of methylation, just sort of global general effects, right? They just wanted to see, if we get rid of this trait, what will happen? So next, the researchers wanted to study how is methylation affected on a specific gene. And what they chose was, I believe, the dynactin 62, IP62 gene. And they used bisulfite sequencing to sort of see what parts of this DNA sequence were methylated. And then they sort of compared results between the control group and the experimental group, between worker bees and queen bees, et cetera, et cetera.
The scientists injected a special molecule known as siRNA into the larvae to reduce the amounts of DNMT3 gene. In Figure A, it shows that the strongest effect of this gene silencing happened about 48 hours after the larvae were injected, which is during the same time that the larvae would be going through a key stage in development as they transition from the L2 to L3 stage. This is similar to the transition from early to late childhood in humans. Another test called in situ hybridization was used to show the scientists where the gene was turned on during early development. The red arrows point to the important area where this gene was being used, shown in Figure B, helping scientists understand the role of DNMT3 in growth. Figure 2 shows how turning off this gene decides whether a bee becomes a queen or a worker. To do this experiment, scientists injected young bees with either DNMT3 or a harmless control gene, meaning that would have no effect, and watched them grow. All of the larvae developed normally, but the adult bees looked different depending on the treatment they received. In Figure 2a, it shows that 72% of the queens with inactive DNMT3 became queens. The larvae who were injected with the control mostly became workers, about 77% of them. In Figure B, it shows that bees who became true queens from the DNMT3 group had fully developed ovaries, while queen-like and worker bees showed less reproductive development with underdeveloped ovaries. Figure C further shows how bees with DNMT3 turned off compared to others, as they had ovaries with the most similar to a virgin queen, while worker bees had smaller and less developed ovaries. Scientists looked at the amount of methylation, a chemical modification that usually turns genes off, of the diactin P26 gene in worker and queen larvae. Queen larvae showed that 48% Queen larvae showed about a 48% decrease in methylation, and worker larvae showed about a 58% decrease in methylation. Figure 3b specifically looked at the heads of the larvae. Worker heads had 73% methylation, higher than their 58% whole body methylation. Queens had 63% methylation, higher than their 48% full body methylation. After the special treatment with the queen larvae head treated with the DNMT3 silencer, they showed 62% methylation overall, while the workers had 79% methylation on a specific gene region.
First, I want to draw your attention to these two graphs. So on the left, we have graph A, and basically what we're looking at here is the control group, which is the group with all their DNA normal, right? And then we have the group where on over here, um, where we knocked out the DNMT3 gene. So these guys don't have regular methylation. And what we can see is that there's a lot more queens coming from the group that had their methylation center knocked out than from the normal wild type group, right? So that was one of the two major findings of this paper. And the second one I sort of want to draw your attention to here is the graphic on the right. So what we're seeing here is that there is a 10% difference in methylation in the B graph between queen larva and worker larva in the head. So basically what the researchers did, they just grinded up a bunch of insect heads and did some sequencing, right? And they looked at these specific site using bisulfite sequencing, and they compared the proportions of these sites that were methylated. And they found that worker larvae had a higher percentage of methylation than queen larvae. And in this sample, we see that um, the RNA knockout group has basically the same 79% versus 73% up here, basically the same sort of, methylation makeup as the worker larva. So what that indicates, here are the main takeaways, just general summary of what I just said. What that indicates is that DNMT3 is somehow involved in this process, but of differentiating these honeybees into different roles. Because the methylation patterns are so similar, and we observe that, getting rid of this DNMT3 sort of led to the same methylation pattern. So this is just a general overview of what I just said. Methylation has a significant effect on honeybee roll determination. Less methylation means more queens. Worker bees are more methylated than queen bees, and the DNMT3 group queen bees showed similar methylation patterns and ratios to the wild type queen bees and the dynactin P62 gene. So, yeah, those were the main takeaways from the researchers. Everything was controlled, positive, negative controls, all made sense. Um, so we could sort of conclude that, yeah, these conclusions make sense.
And going further with a conclusion and where might we go next with this study, scientists have decided that what honeybees are eating can cause them to be queens or workers in a beehive. This is called, by that word, DNA methylation, which is turning your genes off or on. And scientists have turned off a gene called DMT3 in this study, which led to more queens than worker bees in this beehive. This meant that the food that they used called royal jelly in this study specifically decided how the bees will grow, as queens or as worker bees. This is important for beekeepers in Pittsburgh and all of Chatham County and across the country too, as honeybees help to pollinate the crops, and they make the honey, which a lot of families use for their income. If scientists can use this study and can apply the evidence that they learn from it, they can create better bees, and beekeepers could vitalize this to make better hives, which make more honey. And it could also help to solve some of the beekeeping related issues that they encounter, which leads to, hives breaking down. Additionally, this study could also help people with other bugs like wasps and ants, and even, pests that aren't bugs, that, hurt crops and growth. But it could also help people. If food affects bee genes, it could also affect human genes. This could help with human-related gene problems that lead to things like diabetes or memory loss or any gene problem that you may know of. For beekeepers and farmers in the future, this means making better beehives, and making better bees means healthier crops. Beekeeping across Chatham County and the whole country and even the whole world could be improved greatly. This would be more sustainable for crops and the income and just the overall health. So that does it for our conclusion.